excited about the next section?
Very.
You get 2 snippets.
Oh. Oh, Josie.
All right. Where's my shield? Oh, here it is. Let's get mouse. All right.
Did you turn your power off?
I did not. You got me.
Do we need biography groups?
Yes.
I think we do. We probably do.
Yay.
Awesome.
But we're not playing biography today, so let's do them tomorrow.
Oh, so no quiz tomorrow.
Ooh.
You never know.
You walked yourself into that one, Mr. Willis.
Good catch, Joe.
200 IQ.
You don't know. Y'all distract me. Alright.
You're slacking today.
My 12th trip back and forth across the Alright, the first thing we need to do is we need to talk about the structure of DNA. And we already learned this in like chapter 3 or something like that. Okay? So you should already know this. But DNA is made of these things. What are these things called?
Nucleotides.
Nucleotides. I've given you 4 nucleotides. Take a look at them. The nucleotides have, each nucleotide has 3 parts. It has a sugar part. What's the sugar called?
Deoxyribose.
Deoxyribose sugar. And I want you to notice something. It's a 5-carbon sugar. Do you see the numbers? 1, 2, 3, 4, 5 on the sugar there? That's the carbon atoms. The carbon atoms are numbered. And they're always given a dash. That's called prime. So the 3' carbon, the 5' carbon. Those are the 2 ones we're interested in. The 3' carbon is on the, I guess you could call it the bottom, as I've written the words here. But of course, this is just a molecule. It can be flipped around in any direction. But on the bottom of the model is the 3' carbon, and the 5' carbon is adjacent to the phosphate group. You see how the 5' is next to the phosphate? Always remember 5-phosphate. They both start with ph. 5-phosphate. The phosphate's on the 5' end. Can you remember that?
Yes.
You'll need to remember it. And so that's the sugar. It's a 5-carbon sugar. It's the same as like glucose, except glucose is a 6-carbon sugar. So this is a 5-carbon sugar. Sometimes they call it a pentose sugar. Why would they call it a pentose sugar? The pentagon, 5 sides, pent means 5. Pentose. So 5-carbon sugar. And then there's a phosphate group. So sugar, phosphate, and the letter that you see stands for the nitrogen base. Now, there's 2 types of nitrogen bases. Notice the A and the G bases that I've given you are larger than the C and the T bases. You notice that on the model? The G and the A are double-ringed bases. The C and the T are single-ringed bases. And when I say double ring and single ring, look at the screen up here. You see how A, this is the adenine, A stands for adenine, it has 2 rings on the nitrogen base. And so does guanine, has 2 rings to the nitrogen base. But thymine here has only 1 ring on the nitrogen base. And cytosine has only 1 ring on the nitrogen base. Are y'all checking this out? Do you see why they call it a nitrogen base? Look at all the nitrogen in the structure. Nitrogen, 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 nitrogen. There's a lot of nitrogen in the nitrogen bases.
What does that add to it?
It just gives it properties that make it different from other molecules. So the double ring bases, the larger bases, are called the purines. G and A are purines. You know how I used to remember that? People from Georgia are pure. G and A are purines. People from Georgia are pure. That's how I remember it. Isn't that great? It's one great way to remember it. The pyrimidines are C and T. People from Connecticut are pyros. That's how I remember that.
Greta has a good one.
Greta, what's yours?
Cleopatra and Tut live in pyramids.
Oh my goodness, that's way better than mine. Cleopatra and Tut live in pyramids. Greta, that's fantastic. Can I steal that from you? I'll tell future classes that that was Greta's.
That'll be my legacy.
That'll be your legacy.
You should write it on a piece of
Good to have a legacy.
paper.
So, those are the different bases. Now, I want you to notice that the G can attach to the C. If you turn the C upside down, it fits right in there with the G. And the A can attach to the T. But you have to turn the one of them goes upside down from the other one. Y'all notice that? Play with your molecules, get them to attach. And then they fit to one another like this, so that you can make a ladder. That's the double helix formation. Now, the first person to shed light that the G's and C's might connect, that the purines and pyrimidines connect to one another, was a scientist named Chargaff, or Chargaff. Chargaff. Chargaff was a chemist, and he took 6 species of organisms: humans, fruit flies, corn, a type of fungus, E. coli bacteria, and Bacillus subtilis bacteria. He took these species and he disrupted their cells, and he used chemical tests that could measure the amount of A's, T's, G's, and C's in the sample from each organism. And lo and behold, he found that the same amounts of A's and T's always occurred for each organism. Look at humans, have 31 A, T, and only around 19 G, C. It looks like the A's and T's are about the same amount, and the G's and C's are about the same amount. This was a famous experiment. He also won a Nobel Prize for it. And that sheds light on the fact that A bonds to T, and G bonds to C. Watson and Crick saw this data and figured out that those must be bonded together if they're always the same number. Look, in every organism they're the same number. Look at E. coli. It's about the same. Why aren't they exactly the same?
There's another kind.
Experimental error. Experiments aren't perfect. There's also a lot of free-floating nucleotides in the nucleus that aren't attached to one another. A lot of reasons. So they might ask you on the test or exam about Chargaff's data. He was the one that shows that A and T bond together and G and C bond together. His experiments. Make sense? So, if you put, this shows, what molecule is this? You recognize that molecule? It's got a 5-carbon sugar. It does. It's got a base that attaches to the sugar. And it's got phosphate groups.
ATP.
It's ATP. See how it has 3 phosphate groups? ATP is just basically one of these adenines with 2 more phosphate groups. So it has 3 phosphate groups total. So ATP is basically a nucleotide that's a little bit special. Well, we learned about ATP when we did photosynthesis respiration. Remember all that? Mitochondrion. Okay. We talked about this yesterday. Here's the double helix model. Now, I want you to take a look at the models I gave you. You see how the G and C have these dotted lines where the G and C connect together? See how G and C have 3 dotted lines? And A and T have 2 dotted lines? That has to do with the fact that when you join the bases together, the A and the T only join with 2 hydrogen bonds. They're joined, DNA is joined in the middle by hydrogen bonds. Hydrogen bonds are relatively weak compared to the covalent bonds that join the sides of the chain. The sides of the chain are joined together by covalent bonds. That's sharing of electrons. Very strong on the sides. But the middle is joined by hydrogen bonds, which are a little bit more like magnetic attractions. They can be broken. And if you notice, A and T join together with only 2 hydrogen bonds. G and C join together with 3 hydrogen bonds. This is an important thing. There's a difference between A's and T's and G's and C's. G's and C's are connected together more strongly. And that's what those dotted lines are about. DNA, you see, can be unzipped. There are enzymes that can unzip the DNA. But it's harder to unzip it if there's a bunch of G's and C's than if there's a bunch of A's and T's. And when we talk about control of transcription and translation and DNA replication, we're going to talk more about that. But DNA can be unzipped in the middle, or if you heat it up, if you take DNA and you get it in a test tube and make it hot, the molecules start vibrating and it'll just it'll come apart. That's another thing scientists use to break apart DNA and manipulate it, is the fact that if you get it hot, those hydrogen bonds won't be able to hold it together and it'll come apart. Pretty good, huh?
Mm-hmm.
Now, DNA has what's called an anti-parallel nature. See that this strand on the left side here I have is right side up, and the other strand on the opposite side is upside down. We call that anti-parallel. Here's parallel, here's anti-parallel. That's my DNA dance at Ziggy's.
Do it to him.
Did I show you this last semester?
Wait.
Break some more blues.
Woo.
People don't know I'm doing the DNA dance.
Wait.
Pretty good. We would say that the DNA on this side goes from the 5' to the 3' direction. And that the DNA on the other side goes from the 3' to the 5' direction. So notice the 5' is where the phosphate is. And the 3' is on the other side. Wherever the phosphate is, the easiest way to remember this is wherever the phosphate is sticking up, that's the 5' end. So here the phosphate is down this way, so that's the 5' end. Phosphate 5. And so DNA has what's called an anti-parallel nature. And that's going to be important when we're copying DNA and doing transcription and translation. You have to understand that idea. Questions? Hit me with some questions.
Why is RNA sugar different from
Yeah, RNA is a little bit different. And here I have some RNA nucleotides. RNA has a few differences. RNA has ribose instead of deoxyribose sugar. It's a little bit different sugar. It has an extra oxygen atom. So that's why they call it deoxyribose, because the DNA is missing one of the oxygens in the sugar. And RNA has U instead of T. It has uracil instead of thymine. Uracil and thymine are very close as far as bases are concerned, but they're a little different. And you usually find RNA in a single-stranded molecule instead of double-stranded. Although there is double-stranded RNA, but you just don't find it that frequently. And we're going to bring up and talk about RNA in both DNA replication and transcription and translation. So RNA discussion is coming up soon. Yes, Charlie?
Does uracil affect the unzipping?
It has two bonds just the same way that thymine has. So it pretty much unzips the same way. Other questions about the structure of DNA? And of course, it's not just like a ladder that goes on the side of a house. It's a twisted ladder. As you can see in this picture right here, both those pictures, it twists. How often does it twist? Every 10 bases that you count down, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, it would have done one full twist. It does a full twist every 10 base pairs. And so it's like a twisted ladder. If you're climbing up this ladder, you'd be going in circles. Every 10 runs, you do a circle. That's another dance I like.
Wait.
They don't know what you're doing. They don't know you're doing it. If you ever meet someone who knows you're doing that, you're like, I like this chick. We don't have any of those. Different crowd. Okay, other questions? We got a little off topic. All right. Now let's go on to, I want to show you a really cool chemical thing that they can do. If I could. Oh, here we go. I want to show you a process called DNA sequencing. In DNA sequencing, they can use machines to figure out the DNA sequence, the A's and T's and C's and G's. They can sequence your DNA. Now, they first started doing this when I was about your age. They had this big project called the Human Genome Project. And they took like thousands of scientists and they used a process to sequence the DNA and figure out every letter in the human DNA code. And it took like 13 years, and they finished it. They now have the entire human DNA sequence in computer banks. It's 3 billion letters long. If you had a book like this that was just full of letters, no pictures, just letters, and it was all A's and T's and C's and G's, it would take about 30 books of A's and T's and C's and G's.
What's the point of doing that?
For the whole thing.
Why would they do that then?
So once they do that, they could figure out, like, let's say you had an identical twin. And you end up normal, but your identical twin has some genetic disorder. They could go through and they could see what letter he has different from you. And why that's causing the disease. You see what I'm saying?
Yeah.
And they could figure out what disorders are genetic and where the mistake is. And now they've come up with ways of correcting mistakes. And they could actually maybe fix some genetic disorders. There's a lot of genetic disorders out there. So that's just one of the processes. They can figure out things about the DNA. They can do all this DNA technology, like putting making new sequences. We have a whole chapter on this stuff. So that's a great question. Yeah.
What if you don't have an identical twin? Do they still just look?
Yeah, they can still look at your DNA and they can compare you. If you have some kind of genetic disorder and other people don't, they can compare your genes and get clues to why you're different. Doesn't have to be an identical twin, but that would be the easiest to see a difference. But this is what they're working on right now. They're trying to figure out, what every gene does. And they found, very interestingly, that most of the DNA doesn't code for anything. It's mostly kind of nonsense instructions that are ignored by your cells. Something like at least 50-60% of your DNA is not even used. Kind of weird.
Did it used to be used?
They think that it might have come up through evolution, and maybe it was used in ancestors. Like you might have some bacterial genes there that are just turned off and not used, because if you go back far enough, all things came from bacteria that have evolved. And so it's very interesting what's in there. But let me show you how they sequenced it. That's one thing you need to understand. So we take our DNA strand. I'm going to turn it sideways because it's just easier to teach this way. And what scientists would do is get it hot and put it in a test tube, and it would come apart, and we're just going to work with half of the strand. Because as we know, if we know one strand, we'll know the other one, right? Because the T always bonds with an A, and the A and the C always bonds with a G. So all you got to do is know one strand. So they heat it up, it separates, and we're just going to work with this strand right here. This is called Sanger sequencing, by the way. This guy named Sanger figured out how to do this. They actually have other ways of doing it now that are faster, but this is a really cool way. Now they take what they've done, the scientists in the lab have made special nucleotides. Special A's and T's and C's and G's that have been manipulated where they don't have the OH that's connected to the 3' carbon. That OH is missing. And what that does is, when you're building a DNA strand, if one of these special nucleotides, they're called terminator nucleotides, they're actually called DDNTPs, which stands for some long phrase. But I like to call them terminator nucleotides. Most people call them terminator nucleotides. It's just easier to say than DDNTP. Or the longer name, whatever DDNTP stands for. So anyway, these special nucleotides are added to the mix. And another thing that's added is what we call these DNA primers. These DNA primers are 3 nucleotides that have been connected together in the lab by scientists. And I have 3 here. In actuality, it's probably 50. But I have just 3 to be able to show this with my magnets. It's a sequence of nucleotides that's known to connect to the DNA at a certain location. We call it a DNA primer. So for instance, if a scientist wants to sequence this particular gene, they figure out what's at the beginning, and they make a primer that will connect to it. Are y'all following here? This isn't easy. Any specific questions? So, you put the DNA in a test tube, you heat it up, it separates, you put in the terminator nucleotides, you put in regular nucleotides, you can do this all in a test tube. And then you let it cool down. And what happens first is the primer connects to the beginning of the strand. And there's an enzyme called DNA polymerase that they also put in the test tube. I have a DNA polymerase enzyme here. And what the DNA polymerase does is it takes nucleotides and it matches them up to the DNA strand. Click. That's a T, so an A goes with it. Click. That's a C, so it goes to a G. Here's a C here. What would go to that?
G.
G. And this DNA polymerase just comes up here and randomly grabs nucleotides. Well, here's a G. What if it randomly grabbed one of these terminator nucleotides? It puts that down, click, and that stops the strand. Another nucleotide cannot be added here because this is a terminator nucleotide. It's been manipulated, so you can't add any more to it.
Can you add a primer?
We could add another primer, but we don't want to. We just want the primer to stick at the beginning. So we want it to be terminated, in other words. That only takes just a second for all that to happen. And then the scientists heat up the DNA again, and this comes apart. And I'm just going to kind of store this thing over here. In actuality, it's floating around in the test tube. And they do the whole process again. Primer attaches. DNA polymerase goes to work. Click. Click. Let's say we get a terminator right here. Click. We can't make any more. This detaches. We do the whole thing again. Primer attaches. Let's pretend for this one we get a terminator right away. The first one happens to be a terminator. Click. It's just random when the terminator is grabbed, you see. And this thing goes away. Let's do it one more time.
Mr. Wood,
Yes?
how exactly do you see this in a test tube?
You can't see it. It's too small to be seen. It's happening down there. I'll show you what you can see in a minute. This is just happening in the test tube. They heat it up, they cool it down, it just happens. Okay? Let's do it one more time. Primer connects. We start adding nucleotides. We get there. And let's say right here we get a terminator nucleotide added. In reality, they do this over and over again hundreds of times. And what you end up with is a variety of different length partial strands, depending on when the terminator nucleotide was added. Red, are you with me here?
Yes.
Hang with me. You see how this is kind of short and this one's longer? Y'all see that? Question?
Is this how they correct?
No, this is not how they correct. They're just sequencing the DNA. They want to see what letters come first, second, third, that kind of thing. What they then do is they stick in the test tube a little vacuum pump, a tube, that will suck up the fragments. The fragments will be sucked into the tube here. Whoops. Now, at one end, at the top end of the tube, is a machine, which is a laser. They use a laser. A laser. It's Austin Powers. And this tube is filled with agar. You know what agar is? It's kind of like gel. It's filled with a gel. It's not water, it's a little thicker than water. And what happens is, when these pieces get sucked up, the light pieces move faster through the agar. And the longer pieces, the heavier pieces, move slower through the agar. Now this tube's kind of long. So that the pieces, as they're being sucked up here and moving through this tube, they sort themselves by size. The shorter pieces move fast through the agar. The longer pieces move slow through the agar. In this, what happens is this laser, as they go past the laser, here's the shortest piece. As they go past the laser fires energy, and it hits the special terminator nucleotide. Now, if the special terminator nucleotide is an A, it will hit it and make it fluoresce green. It'll make it glow green. And there's not only a laser here, there's an electric eye that can see the fluorescence from a single molecule. And so the laser shoots this thing as it's coming by. Nothing really happens here, but this terminator nucleotide will glow green. And the computer can sense that, and when it sees the green glow, it says, oh, that's an A. And then the next longest piece goes through. So that piece has gone through, and the laser measures an A. And then the next longest piece gets there, and that glows purple, and the laser sees a C. C's glow purple. I wish I had a purple marker. And then the next longest piece comes through, and that's the third nucleotide is the next longest piece, and that registers a G. You see? Because it glows yellow. G's glow yellow. These are the actual colors that they glow. And then the next longest piece goes through. It'll just keep getting next longest piece, next longest piece, next longest piece. And so that glows purple again, so that's a C. And so very quickly, now these things are coming through quickly. And it's not just one piece that comes through. There's hundreds of little pieces, hundreds of these size pieces, and hundreds of these size pieces, and hundreds of these size pieces. So as you can see, what they actually get is a curve. So the first piece here the first piece here was G, and the next piece is this peak means there's the higher the peak, the more came through. So there were first a lot of G's came through, then a whole lot of A's came through, then a whole lot of T's came through, and so forth. So they know the sequence of the DNA from one side to the other. That's called Sanger sequencing. These pieces here were real long, they came through later. These pieces here were real short, they came through earlier. Isn't that cool?
Mm-hmm.
That's how they sequence DNA, or that's how they used to do it. They have even faster ways now. They can sequence an entire genome now in just a couple of days, where the first one that was sequenced took 13 years. And thousands of scientists. Now they just have a machine. They take the DNA sample, they stick it in the machine, it does it, and you get your answer in just a few days. Depending on the organism, it could be just a few hours. They can sequence viruses and bacteria just very quickly now.
What do they do for ancestry.com?
I think that's gel electrophoresis. This is a different thing we're going to talk about in a later chapter, where they separate DNA and get these bands, and that can also tell you about pieces of DNA and how well you match up with others. And that's not actually getting individual A's and T's and C's and G's, but that gets each band you can think of might be an entire gene. And so they can compare similar genes between people and come up with where you were probably born and that sort of thing.
Crazy.
Any questions about Sanger sequencing?
These are wipers.
I meant to show you this video.
What do you want us to do with these?
Hold on. Let me see how long this video is. Actually, I'll tell you what. I'll show it to you next time. Let me get the pieces. Put all the DNA pieces right here on this little shelf. And did we get wipers yesterday?
Yeah, you gave me a lot of